Good afternoon, everybody. I'm very happy to welcome you all to today's Kaapi with Curiosity. As many of you know, we have many programs going on for popularization of science and non-formal science education. Today I would like to talk just about one activity, one program which is happening day after tomorrow on Tuesday. Tuesday in Bangalore, it is going to be Zero Shadow Day. That is at local noon, vertical post will not cast any shadow. And this has a historical importance in the sense that some 2000 years ago, Greek scientist Eratosthenes used this fact to calculate the circumference of the Earth. So we have lined up many activities on that day. One is we have a workshop for students where they come here and then see that there is no shadow in Bangalore. We have associated with Aryabhat Foundation in Bhopal, who will be measuring the shortest shadow cast there. And they will be giving us that information. Children will use that information and calculate the circumference of the Earth. Also, we have public lectures on that day by my senior colleague, Dr. Shailaja, and Professor P. Venkatakrishnan, formerly director of Udaipur Solar Observatory. We also have many demonstrations of this event. I invite you all to participate in these programs. Kaapi with Curiosity was started almost a year and a half ago, and we have had many of those lectures here. It is becoming popular with every passing day. Many distinguished scientists have given lecture here. And today we have a very eminent scientist, Dr. Zorana Zeravcic, who will talk about Towards Life in Jar. Hearty welcome to you, madam. Warm welcome to all the distinguished guests here. I request Professor Rajesh Gopakumar to speak a few words about ICTS. Thank you.
Thank you, Dr. Pramod, and welcome everyone. It's indeed a pleasure to have you, Dr. Zorana, here for this particular event today afternoon. I'll just say a few words about ICTS, though I feel increasingly that it's superfluous since I see many of you here in our events month to month, so presumably I'm boring you with repeating the same stuff about ICTS, but just in case some of you who are here for the first time, ICTS is the International Center for Theoretical Sciences. It's a new and for us very exciting institution in the north of Bangalore, in the northwest of Bangalore. We are different from most other research institutes, of which Bangalore has several, in that we have not only our own research, which covers a number of areas in theoretical physics, mathematics, and increasingly in quantitative biology, and going ahead even theoretical computer science, but we also have linked in a very close way with our own research are the programs that we organize, which consist of meetings with the intention of bringing researchers from around the world together with the Indian community to collaborate, to discuss, and also to sort of train some of the younger students and postdocs of India in exciting new areas of science worldwide. So that's one of the other main components of our mission, so not just our research, but also the programs. But the third component of our research, which is also integrally tied up with the other two, is outreach, and which is why events like today's and Kaapi with Curiosity is one of our flagship events, and we have been running it, as Dr. Pramod mentioned, from October 2016. I mean, we've had a great response from the Bangalore lay public, and we've had a number of speakers, both who have come for our programs, like today. Dr. Zorana is here as part of part of the Living Matter program, which we are having at ICTS last week and the coming week. So Kaapi with Kuriosity is one such regular outreach event that we have, but we also have public lectures at ICTS. We had some very successful ones earlier this year, including by Kip Thorne, last year's Nobel laureate for gravitational wave detection. So there are a number of events there. Next month, I think we'll have one by Juan Maldacena on black holes and the structure of space-time. So we organize transport from the city, including from the Planetarium. So, so those of you who haven't been to our campus, or even if you have been and would like to, please come for the public lecture on campus. We also have a number of other outreach activities like the Einstein lectures, in which many of people from ICTS and visitors go to some colleges across India. So this is just a snapshot of some of the things that we do at ICTS. Yeah, so let me just conclude by saying that it's great to have this lecture in collaboration with the Planetarium. It has been a very successful partnership, and I hope this will continue in years to come. Before we invite Vijay to introduce the speaker, I would invite Dr. Nanjundaya to maybe hand over a memento to the speaker on behalf of ICTS. So Vijay will introduce the speaker.
So it's a great pleasure to introduce Dr. Zorana Zeravcic. So Zorana did her early studies, undergraduate studies at the University of Belgrade. She did a PhD at Leiden University. She then moved to the US, did a postdoc at Harvard University, and then one more postdoc at Rockefeller Center. She moved back to France, and where she is now an assistant professor at the ESPCI. Zorana is one of the rising stars in trying to understand artificial life. She focuses on how we try to understand the fundamental principles of the building blocks of life, how emergent structures can come about from simple replicating systems. It's a fun thing to read her papers and her blog and try to understand, not only explain what is possible in life, what are the other things which are possible with the fundamental physics that she looks at. So we look forward to a great talk by Zorana.
Thank you, Vijay, very much. And it's really a pleasure to be here and to give this Kaapi with Kuriosity lecture. And so as Vijay said, my name is Zorana, and I work at this place called the ESPCI. So it's a school located in Paris, somewhere around here, so it's not far from Notre Dame and from the Latin Quarter. And this ESPCI is just really a very long acronym that is written here, which basically means it's a school of higher education for chemistry and physics with industrial application. Now, the school was founded in 1882 by the city of Paris. It has free education, but what is very interesting about it is that it is an interdisciplinary education. So from the very founding, it was teaching physics and chemistry to students, but in recent decades, biology was also added. And all of the students actually take all of these classes from all of these subjects. Now, you may have not heard about this school, but maybe you've heard about one of her most famous alumni, which is Madame Marie Curie. So she's a famous chemist and a double Nobel Prize winner. And she is the one who was responsible for the discovery and coined the term radioactive and radioactivity. And she was the one who first observed how, if you expose tumor or cancer cells to radioactive elements, they can sort of shrink or even decay. So in many ways, she influenced the modern medicine, but also all of our lives in some way. Now, nowadays, at ESPCI, we don't work on radioactivity anymore. Maybe there is an occasional radioactive doorknob somewhere, but we don't do this research anymore. But we are still very much inspired by her spirit and work on fundamental problems. that are in physics, chemistry, and biology, but also at the interfaces of these sciences. And this is why I love this school, and this is why I'm there, because I'm trained as a physicist, but I'm absolutely fascinated by biology and the things that I'm interested in, and really interface of these two sciences. So when I think about biology and living things, what fascinates me is that they are just made out of few types of building blocks. Very roughly, these are carbohydrates or sugars. So what I'm showing you here is a lactose molecule. So this contributes to the sweetness of the milk you're drinking. Then another building block is water. It's all around us, it's in us, right? We are 3/4 of us is just water. Then we have DNA, or nucleic acids. So these are these very long molecules that have information about you, inside you. They know all about you. And we have something that are called proteins that are made out of amino acids. And there are 20 amino acids that make up all of the proteins that are in the living systems. And this is an example of a protein called hemoglobin that helps transport oxygen in your blood. So these are a few building blocks. And the interesting thing is if we take them and mix them together, of course, we are not mixing them together. Nature has done this over eons. We end up having all these interesting things here. Beautiful vegetation, human beings, these very cute echidna, and these very colorful medusa. These are just examples. And now, I have a question for the audience. Well, actually, two questions. So first question is, imagine that we go all around the Earth, and we collect all of the different types of trees we can find, and we collect them in one space. The question for you is, so all of the ones that exist on Earth, the question for you is, if we do that, are we going to see all the possible trees that we can actually make from the building blocks that comprise living things? Okay, there are some no. There is a clever, yes?
I don't think so, because the combinations are too much to make all the trees which we collect from the odd combinations.
Okay, we have a scientist in the making. Okay, thank you. Indeed, the answer is no. We are going to see just some things, okay? And then the other question is, imagine that sometime in the evolution of Earth, instead of rain made out of water falling down, we had lemon juice falling from the sky. Would we actually see the same type of medusa that we see on this picture? Yeah, so we hear no as well. So this is exactly the thing that is very interesting. So although very broad and diverse, what we actually see when we look outside is just a small set of all the possible things that we can actually make out of the building blocks that I see. And they're products of some either accidents or some solutions that were good at some point in time in the evolution of Earth. And now, I told you I'm a physicist, and as a physicist, when I look outside, what I'm really interested in is, can we find what are laws and rules behind what we see in nature? And there is this one quote by scientist Christopher Langton that I like very much, which says, to be able to have a theory of actual, so to know all these laws and rules, it is really necessary to understand what are all the possibilities that we actually have. And this is what very much inspires me. And now, maybe this sounds a little bit strange. We want to know all the possible things, and why is it necessary to know all the possible things? But I will give you one example of a field that you already experienced, did something like this, and I will tell you what it gave us. And this is the field of chemistry. Okay, so it's a very old field, and the way it started is that the chemist, went out, saw what is there in the nature. They started looking at these different compounds, and they wanted to understand what they are made of. But only when they broke them apart to building blocks and then tried to combine them in these building blocks in all all the possible ways did they understand how things actually function, and which led to rules and laws that are now comprising the modern chemistry. Because without that, if they hadn't done that, we wouldn't have things like synthetic rubber or plastics, things that are all around you. They wouldn't exist, because this is not something you see in the nature. It's something that you don't see in the nature. So it's very important to understand, to be able to build these laws and rules, to understand what are all the possible things. And so this leads me to this field, a relatively young field that is called artificial life, whose goal is to really look at the building blocks that exist in nature, but also ones that don't exist in the nature, and try to explore what are all the possibilities of creating things. And one of the main dreams of this field is to, first of all, learn about biology. And I will give you an example of that. So here at the bottom, you have a caterpillar, right, that is just crawling on some part of the wood. And here on the top, you actually have a simulation where scientists took building blocks, which are cubes. Some are squishy, okay, so they can squish like muscles. And these are the blue and the red ones. But there are also some that are hard like bones. These are the green ones. And they took many different combinations and compositions of these different blocks and put them together to make objects, okay, to make bodies. And out of all the possibilities that they looked at, they selected those that actually show some sort of a motion, okay? And so one of the things that they found is that, indeed, if you have these types of building blocks, to efficiently actually move, you can actually crawl like a caterpillar. So they found out that this is a very efficient way to actually move. So nature did something very efficiently. But they also found something that is not seen in nature. And this is an example that I'm showing you here. It's a very interesting one, because this object moves very efficiently, as efficiently as a caterpillar, but it actually shakes around and moves. And this is not something you typically see in nature. Which leads me to the next dream of this field, and this is the dream of discovering new things and new properties. Now, physicists like to show this map, which is an ancient map of a naval map. So this is a coast of Sweden, and it's supposed to depict this sea, which is filled of creatures that we don't know about. And the goal of explorers is actually to travel in this sea and try to find what are these creatures and to discover new things. So really, this is one of the dreams of this field. And then the last dream is, the last big dream is that if we actually learn about these things and discover new things, we'll be able to actually improve industry and technology the same way that chemistry did in the example that I gave you. And there are already some steps in doing that. So what I'm depicting here is a textile shirt that is designed, inspired by biological, it's a plant that eats animals, small animals. And the skin of this plant is such that it repels water, okay? And so the scientists were very inspired, and they figured out how to make this type of skin. And now they can actually make shirts that completely repel water. So if you just pour the water, it won't get wet, okay? And this is just one example, but we can imagine all kinds of things that are inspired by living systems. So materials that can self-replicate, that can change and adapt depending on the environment they're in, and so on. And any futuristic material, basically, you can imagine building them. And this is all very nice, of course, but there is a huge disclaimer here. Okay? So on one hand side, you can imagine in the future, if we actually manage to discover new things and to make new artificial things, artificial living matter, there is going to be a huge question about somehow classifying these things, ethical questions of whether trying to define what actually life means, okay? But there is also another very big issue that we have to keep in mind all the time, and this is that as we advance and make these functional, interesting things, it's very easy to imagine them being misused and abused. So I'm not going to try to define life, and we are not there yet, so we are not that advanced, but it's very important to try to keep in mind that is what we are doing, it's although very interesting and very exciting and very important, we have to keep in mind that there are these dangerous things, dangerous borders that we should not cross. Okay, so this is the field of artificial life, and as I said, I'm not going to define life, and I'm just going to tell you about how we are trying to mimic things that we see in nature. And to do that, I first have to tell you a little bit what are the main ingredients of how nature makes things. So there are the way that we make things, people make things, and the way nature makes things are very, very different. Okay? So these are two relatively analogous things. So here at the top, you have a computer. So it's one of the most advanced things a man can make. And here at the bottom, you have something that's called a ribosome. So it's a very complex molecular machine that is involved in protein synthesis. So your body has tens of trillions of cells, and each cell has like tens of millions of these little things inside. So they're very, very important for you. Now, both of these things are made out of building blocks. On the top, we have a computer. It's made out of the circuit board and all the little diodes and CPUs, etc. And here at the bottom, we have the RNA, which is the central part. So it's a long nucleic acid, the central part of this machine. And then the rest of the things are proteins that are made out of amino acids. We mentioned them already. So there are 20 amino acids. And now, the interesting thing is, to make any of these things, we need some sort of a blueprint, a scheme of how things should be made. And now, on the top case, this is literally a scheme which tells, a robot which component to take and where to put it. And in the bottom case, it's not very visible, but you basically should see here a squiggly line going all around. And then there are these red pieces, which are places where these different proteins should go. And now this is really necessary because if you ever played with Lego pieces, for example, and you have this an image of a ship or a boat that you want to build, you really need an instruction that tells you which piece should go where. It's very hard to build it without this blueprint. And now, so although these two things are analogous, the way that they are made is very different. So in the top case, we use what is called a top-down approach, so an assembly line approach. So you have a robot that takes these pieces and puts them together. And here at the bottom, what nature uses is called the bottom-up approach, or the self-assembly approach, where these components spontaneously come together to form the ribosome. So these things just float around and just spontaneously attach to the right places and form these very complex and functional machines. But not only that, of course, what we here have on the bottom is a much more complex functional thing because it can also repair itself, replicate, adapt, and evolve. And so if we want to mimic what nature does, it is very important to understand this process of self-assembly, okay? But it's also very important to understand what I call specific interactions, and I hope this will become clear in a few slides. Okay, so this is what we want to understand, and now, because we are, first of all, theorists and physicists, we need a model system where we are going to try to understand and to mimic what nature does. And the system that we are going to use is a system of spheres. We love spheres. Physicists love spheres. And if they ever told you they don't like spheres, that's a lie. We all just love spheres. And if you can just make and explain everything by using spheres, we would definitely do that. This is the case for us here as well. The only thing is that our spheres are going to be micron size. So you cannot see them with your eye. You can maybe feel them between your fingers. And we are going to take them and immerse them in a sea of even smaller particles. And these smaller particles are very important because they're very, very excited and active, and they're going to push our big particle around. And so are there any children in the audience? Raise your hand. Children less than 10. Yes, okay. Okay, we have some fake children over there. But, okay, so it's going to be very important. Children, remember that we have these small particles because they're going to be crucial for an experiment that we will do in a little bit. But in any case, so our model system is basically spheres that we take, micron-sized sphere, and put in a jar. And because of these small particles around, these spheres move around, and something interesting happens. And so I hope you can actually see this. So this is a movie from an experiment of my colleagues. So basically what they did, they took 6 spheres. They're roughly all identical. They've put them into this little jar of small particles. And what happened, and I'm not sure you can really see it, but you have to believe me, these spheres came together and formed a structure. So basically, the process that I showed on the previous slide for the ribosome, where things spontaneously come together and form the structure, is the process that we have here in this experiment as well. So these particles spontaneously come together and form structures. So we have a system that can self-assemble things. Okay, so this is the first step. This is very important. Okay, now we took 6 spheres. If we took 7 or 8 or 3, we would get all kinds of different structures. And just to demonstrate that, we're going to do a live demo. So I need 7 volunteers and 3 kids. And I'm not going to pick volunteers. I just expect 7 to show up here. I only want 7. Exactly, the first 7 here. Okay, you belong to the kid category. I need the kid, 1, 2, 3 kids. Okay, so we have enough kids. 2, 4, okay, perfect. So, we are going to demonstrate this self-assembly process. So we are going to be particles. And can you put, please, this around your head? Okay. I should have, okay, I have one more, just 1 second. Okay, here. Here we go. Okay, so you're very important, and you're crucial for everything that's going to happen and everything that's happened in biology, but you'll stay here for a second. Okay. Yes. Okay, so all of our volunteers have this sign around their necks. So it's basically saying, I'm a blue particle, and I like blue particles. Now, I showed you a system of spheres, okay? But we people are really not spheres, so we cannot just bind anywhere. We're going to be binding using our hands. But our hands, just like blue particles, it doesn't matter how. So, you're our assembly system, and I just want you to assemble. You have 2 binding sites, and you can assemble. Okay, this was an obvious choice. Now the temperature, or the kids, or the small particles, break apart their hands. They did it in a wrong way. Okay, now children come here, break apart. The children can move, so they break bonds. Now you broke bonds, okay. No, you broke bonds, you move around. Now you can again make bonds. You can bind with anyone. You don't have... Okay, so we first had a circle of 7. Now we have a circle of 5 and a pair. Now break hands again. Break everywhere, and then come back here. And now move around. Okay, ooh, now we have a very complicated one, but 3, 3, and we have one that is not bound. We can break again. It's actually a fun game. you can do. You can break again, yes, yes. Break again, what else can happen? Yes. Yeah, you can, sure, yes. Okay, there you go. Ah. So now we have 2, 2, and 3, but if we have very agitated little particles, we never might bind anything. But I thank you very much. But yeah, you can just let it down, it doesn't matter. But the purpose of this demonstration was to show you that, if you have these identical building blocks, and they can bind on 2 places with anyone that they have around, they can bind in many ways. We had 6 in one circle, we had 7 in one circle, we had 3 and 3 and 2, and the very important part were the little children, the excited particle that would break apart things and allow them to assemble again. Okay, so this is how self-assembly works if you have identical building blocks. Now, the same thing happens with our spheres, right? We can take spheres, we can put them together, make them self-assemble, and look at in how many different ways, what are all the possibilities that can happen with taking 2, 3, 4, 6, 10, and so on spheres. And this is a very interesting problem per se, for itself. So physicists and mathematicians have been obsessed with that. And for example, it's very interesting that if you take everything from 1 to 5 particles, there is only 1 structure that can assemble. At 6, there are 2. At 7, there are 5. At 14, there are already 1 million different structures you can assemble. And it goes very rapidly. And this is the same movie that we saw before, again, just of 6 particles being assembled. And here I'm just showing a few from the category of 8. And so what I'm telling you is that we have a nice system where things self-assemble, but if I take 14 particles, I have, 1 million possibilities. And if you remember our ribosome, the thing that we always have in the back of our mind, it's made out of 70 or 80 building blocks. And they always come and make 1 ribosome. They don't make trillions, they make 1. So the question that you have is, how can we just make 1 out of all the possibilities that we have available? Okay? And to follow what nature does, it means we need to take our building blocks, to think carefully about what our building blocks should be, and we need a blueprint, okay, of how things should assemble. And you've done this many times, because you've all solved puzzles, okay? So what I'm showing you is basically just puzzle pieces that are designed in such a way that they assemble an elephant, okay? And now, of course, if you know the alphabet, right, there is no problem for you to assemble this elephant out of these building blocks, because you know that first you start with an A, B goes after A, C goes after B, and so on. So that can help you assemble this. But if you want to give this puzzle as a present to someone who doesn't know alphabet, you need to give them an instruction, right? You need to give them a blueprint of how to do that. I mean, they can try lots of possibilities, but it will take forever. You want to help them. And the way that you help them is by making a blueprint that looks like a table, around, like this one shown here. So it's a table where I just marked partly the alphabet pieces. And basically what we do is we look at the final structure, and whenever 2 pieces are in contact, okay, we put a blue square. If they're not in contact, we leave it white. Okay, so let's just see if we've done it correctly. So A is in contact with B, so A is in contact with B. B is in contact with A and C, and so on, okay? And so this is an example of how you can make a blueprint for how a structure is being made. Okay, so these are puzzle pieces. Now we want to go back to our spheres, okay? We want to build our 6-particle cluster. And just copying what we did with the elephant, we can say, so let's make all of the building blocks different types. Okay, so there are 6 different types of particles. And this is our table, okay? And we look at the structure and then fill the table. So green and green, we just have one type of green. It doesn't like the other type. Green likes yellow, light yellow, pink and red. These are the 4. The other green likes the same. Yellows don't like yellows, but like the rest, and so on. And so this is now our blueprint. Now there is also something very exciting about this blueprint, and I would like to ask your audience, can you see something interesting happening here? Can you observe something here? Okay, yes, it's symmetrical. Again, something that physicists love, mathematicians as well, but physicists especially. You have symmetry somewhere. We are the happiest people on the planet. Exactly, it's symmetrical, which basically means that we don't need 6 different building blocks to make our structure. We can actually only do it with 3, right? Because we have 2 and 2 and 2 columns in a row that are the same. So if we apply the symmetry, we end up with a blueprint that looks like this. So we have 2 types of green, 2 of red, and 2 of yellow. And this is really the blueprint for our structure that we want to build. Okay, and so now that we have that, the goal is to take this, put it in our jar, and see what happens. But I've been really just coloring these particles different colors. I mean, if you think about an experiment, you don't just color them and they know exactly what color they are. You somehow have to specify more carefully what these colors actually are in experiments. And so the question is, how do we do it in experiments? And the answer is we use specific glues that are made out of DNA. Okay, so just focus on this part of the slide for the moment. So it's not the DNA that I can take from any of you. There are actually labs and companies which can make DNA on demand. So basically, again to remind you, DNA is this long polymer that is actually made out of 2 strands. The building blocks that make the DNA are just 4. We call them A, T, C, and G. This is how we mark them. And the 2 strands that comprise the DNA we call complementary strand. It's because there is a blueprint by which these building blocks follow that says that A interacts with T and C interacts with G, and they don't like the other ones. So to make a double-stranded DNA, you take one piece that has one sequence, like here, you just make a complementary sequence, and when they come together, they will bind and form bonds, okay? And so there are these companies where you can go online and say, I want a DNA strand that is AAAAT, and the complementary one, and then they will send us AAAAT and TTTTA in 2 different jars. This is what we actually do in experiments. We make these kinds of orders, and we take these DNA strands, and using some clever chemistry, we glue them to the surface of these particles. And all of a sudden, we can actually color them in different colors, because when 2 particles come together, they're not anymore identical. They have this DNA glue on them. So if the DNA glue is complementary, they will bind. If it's not, they will not. And another very exciting thing about this whole system is that if you just have a strand that is of 11 bases long, so 11 of these letters, you can make over 2 million different building blocks, okay? And they just linked 11. Look at the periodic table. You have 100+ atoms, right? If you make artificial building blocks, you can make many, many more. And this is really what allows us to to dive into this field of artificial life, artificial living matter, because we can just take in any building block we want, we can imagine, we can actually make and try to explore the possibilities. Okay, so, and again, I'm just showing you some cartoons, but this is really what's done in experiments. And I'll show you two examples. So one is our example of the 6-particle structure that we want to make. This was the blueprint. Now, I think that you might not be able to see the red color, but you will see yellow and green. So we started with just 6 particles that were floating around in a jar, and they spontaneously come together, and they formed a structure. But the structure followed this blueprint. So when 2 green ones came together, they didn't bind. When 2 red ones came together, they didn't bind either. But the green and red, when they came together, they had complementary strands. They bound. The same goes for yellow, and so on. So by designing our glues to follow this blueprint, we were able to make exactly the structure that we wanted. And this is another example. I'm not showing you a blueprint, just not to confuse it with the one that we are going to be using throughout the talk. But this is a different flower-like structure. It has 6 different types of specific glues. So this table is 6 by 6. And you start with these particles, and they just come together and form this beautiful structure. And also, it's interesting that these 2 structures, when you make them out of identical particles, it's very actually hard for them to get assembled. It's not easy. You can ask me after the lecture why this is the case. But if you put specific glues, we only get them. And this leads me to the next live demo. We need 6 volunteers. 6 volunteers. Okay. So, we had a demonstration in which we had all of the particles be the same type, and we saw that there were many, many different things that can happen. Now, we are actually going to add some specific glues to our hands. So we cannot bind just to anyone, but we have rules. Okay, so all the participants are carrying one of the 3 different glues. And so what do they say? I am a purple particle. I like, on my left hand, I like red. On my right hand, I like yellow, and so on. So you're in your jar now, and you're supposed to find your partners and to see what can happen. So please assemble. Of course, this one is much harder, right? And it's harder for a reason. Because in the original case, everyone was the same, so whoever you see and grab, it's fine. But now, when you come together, you can only bind if you're complementary. So this takes a little bit longer. Okay, penalty. You cannot have 2 hands. Oh, it's true. My bad. Yes, this is one combination. No, sorry, you cannot. You all like red particle. You're not, so you just... But purple particle. So you can bind here. No, the other way around was correct. Sorry. Okay, so this was, wait. No, here. This was okay. The original one was okay. But you still have room for more. You guys bound, okay. Okay. No, don't break. That's okay. And you have a free hand. You like a red particle, you're red. And you like purple, and he's purple. Okay, after some tries, we got 6 particles in a ring. Is there something else we can get? Tough question. So we have kids that break things apart. What else can we get? Well, this is very clever, yes, you can exchange. If you wear the same tag, it really doesn't matter where you are. This is true. So this is really a scientist in the making. But we can make one other thing. Yes. Okay, so I will be I will separate you a little bit. Yes, you can do 3-3, exactly. You can make, I think the arrangement is not, but you can still make 3-3. So you can make 6, but you can also make 3-3. So it's far less possibilities than we had before. It's still 2 choices, but far less than we had before. Before we had many, many more. So this is the importance of specific interactions. But now, we actually need 3 more participants because we really wanted to make just one structure. Okay? And now I need my colleagues from the Living Matter workshop. Okay. Okay, so you can break apart. Our 3 come here. So they are special. Oh, did I mix this up already? Oh, shoot. Wait. What was that? Okay, like here, and stand there. Next one. So it's harder than it. Okay, here, and you bind here. Okay, and the lovely lady here, and bind here. And so what I have here is what are called enzymes in biology, or catalysts. So there are clever helpers that help us actually assemble, that help nature and biology assemble things. And so we're going to move in the center. There you go. And we're going to put our hands in front. And so now you guys, it's not important for the moment what you have on the side. Just look at what particle are you, okay? And so what they have on their tags is what particles they can bind. So they can bind yellow and red. Okay, I did make a mistake. Okay, sorry. Wait. Yeah, I made a mistake. Sorry. Okay, so how does the biology do this? I really don't know. You're definitely a purple particle. Okay, but you won't bind their hands. You're just going to stand in front because you actually want to form a structure with your hands. So these are just your guides. That's your guide there. We need a yellow one here. Who is yellow? We need, yeah, we can. Okay, you can stand here. No, don't hold hands. This is just your guide. We have a purple, and we have a red. And now you bind hands. But with your colleague particles, not with the catalyst. Catalyst is just your guide. Yeah. Okay. Go. Exactly, and bind hands. Okay. Biology does it in a much more efficient way, but it doesn't matter. The thing is that by following instructions from this enzyme here, they made the 6th circle. And there is nothing else that can be made. They cannot make 3 and 3. They cannot make anything else. And even if you had more particles, and I was an extra purple particle, I wouldn't be able to go anywhere. because the catalyst made what we needed to make. Okay? So this is how we got our one structure. Okay? Thank you very much. Okay. So, what I tried to show you so far is the importance of the process of self-assembly, the importance of having specific interactions between building blocks, and the importance of something, of these enzymes or catalysts that help us do things, okay? And now for the rest of the talk, the rest of the few slides, I will basically tell you a little bit of the science that we've done in the last few years. And really, by following these rules and trying to understand how we can build different things, we were able to mimic efficient self-assembly that exists in biology. We were able to make things that self-replicate, have some metabolic kind of behavior, and adapt a little bit. But what I will just focus on is the process of self-replication. So process of making copies of yourself, or objects making copies of themselves. And this is really an interesting process because, so on one hand side, right, you can, in a very efficient way, make copies of objects, but it's also, if you have a system that efficiently makes copies, you can imagine changing some things at some point in generations and see how this impacts what actually happens, okay? So, questions behind understanding what are the necessary ingredients for an artificial object to copy itself is really old, and really one of the first big thinkers behind this process is a great mathematician, John von Neumann, and he's really the grandfather of digital computers. Without his work, who knows how far we would be in the development of computers. So, and he really just was interested in the question, what are these ingredients? What ingredients do we need to make a machine, make any other possible machine, and then as a special possibility, make a copy of itself. And so he had a list of lots and lots of rules. And so we are going to be very much inspired by von Neumann and ask the same question, just in our system of these colloidal particles coated with DNA. And before we do that, we need to understand this process of templating, and I will just remind you of the DNA replication. So a process where one DNA double strand makes two, so there are copies being made. So as I said, so it's a very long polymer made out of two strands. What happens is that there is a machine called polymerase that comes and separates the two strands, and then takes pieces, building blocks, from the surrounding, and puts the complementary building blocks, of course, one next to each other, and make copies. So the blue is the original strand, and then the red are the copied ones. So basically, it's a templating process. You follow the rules that are on the blue, and just attach complementary building blocks to it. So we're going to do something in this spirit very much. And just to tell you that in recent years, there have been successful experiments where people have designed self-replicating systems. So on the top here, you have the designs, and on the bottom are images of experiments. And you can see there are lots of errors. So they wanted to get two types of circles, like the ones that we just made, copy themselves. And they were successful. And you see there are lots of errors. Here, they were just copying linear strands like this, and they were also successful to some extent. So there has been progress in recent years since von Neumann, who was from 1950s. Nowadays, there are some actual experiments. But what we are going to do is use our colloidal system to do this. But now, before I actually show you the blueprint, how we designed the blueprint, I just want to show you the idea behind this templating process we do. So we basically want to copy these two structures here. So our 6-particle structure, and there is also a 2-particle structure here. And the idea is we take our structures, we immerse them in a sea of particles, and these particles attach to the surface of our structures. Once they are attached, they form bonds in the very similar way we've done with the catalyst, right? Things come in some place and then start forming bonds. And once there are enough bonds formed between these gray particles, they will detach from the parents, and the end product is supposed to make our desired structure. And this might be a little bit confusing, so we'll do the same demonstration, not in live, because we can not make people climb on people, but we're going to do it using magnetic toys and ball bearings. Okay. So, we started from our octahedral structure and their dimer. It's immersed in a sea of particles. They can attach one particle per particle in the solution. So this was the second parent. Of course, biology does this in a matter of milliseconds, but okay. We are not there yet. Okay, so now we have two parents with attached particles, and now these particles are attached. They can form bonds between themselves. Again, they all follow some rules, some blueprint, and I will show you in a few minutes how the blueprints look like. So what can happen is that you end up having two triangles on opposite sides of our structure that is called an octahedron. But we want to get an octahedron, not triangles, okay? So we need the little kids, our temperature, that will break bonds. And so things rearrange a little bit. And now we have two particles on one side and four on the other. But this is why we have the second parent that comes with extra two. Okay, and these extra two come with our four, and all of a sudden we have 6 particles together. Okay, and now we're going to say poof, and the green bonds will disappear. Okay, and let's see what we got. We have the original structure of 6, original parent. We have the second parent of 2. We have our, in this case, red structure that has 6, but it's sort of floppy. But what can happen is that this flips, and all of a sudden you get a copy of the first parent. And we have a copy of the second parent. And so where does the copy of the second parent come from? It was here on the side of the first one. Okay, so this was a templating process with our particles. But I was not showing any colors. The actual design looks much more complicated, and this is the blueprint. And you don't need to understand this, but the main thing that I want to tell you is that now designing this blueprint to have a more complicated process that we see in biology becomes, of course, more complicated. And there are also some colors here, so it requires some thinking on how to design this actually. And we don't have experiments in this yet, although they are done, but we have what we call numerical experiments. So this is just a simulation that is calibrated to mimic the experiments that already exist. And in this simulation, we just started with 2 parents and 8 particles in a box. And they followed the blueprint from the previous page. They attached to the parents. You don't see these attached particles very much because I deliberately didn't color them. Otherwise, you wouldn't see the parents. Now you can see these things moving around a lot. You know, these were the kids that are kicking everything around. And after a while, things separate, and you get a copy of the original structure and the copy of the second one. This is a very small simulation. We, of course, do much larger simulations. This is just a snapshot from one of them. And here I'm just trying to show you that we can design these blueprints and interactions so that we get efficient replications, so that very rapidly we can start from 1 and get to 16, 32, and so on. And so this leads me to sort of the end of my talk. I didn't show you, I just showed you one thing that we sort of did, but by trying to mimic nature, we are learning how we can build these blueprints that allow us successful assembly of things, that allow us to build functions, allow us to make things self-replicate. But of course, the dream is that we will learn more and more by trying to mimic the nature and eventually even evolve things and discover new things. And it is very important to know that what we are doing is really at the interface of all these different sciences, and contributions from all of them are very necessary for us to be able actually to do this. And what I showed you, of course, I didn't do by myself. There are lots of colleagues from the US, the Netherlands, France, and more recently from India that are working on this and trying to develop these ideas. And so I thank them, and I thank you for your attention.
Thank you, Zorana, for the wonderful talk and also bringing down the rain, and also showing the true spirit of Kaapi. Doing science is fun. That's the first thing.
Oh, absolutely. I'm not doing it if it's not fun. That's for sure the case.
Okay, so we are open for questions. So there are 2 microphones at the 2 ends. So if you're close to them, please go there. If you're stuck somewhere in the center, just raise your hand. One of our volunteers will come. So there's a hand up there.
So you talked about the blueprint. Is this blueprint is the laws of physics and chemistry, or extra something? Or is there any hierarchy of blueprint? Like there is, like DNA, it encodes all the proteins or all kinds of things. Then there is a hierarchy, then it controls the blueprint of other thing, and then it controls the blueprint of other thing.
Okay, so thank you. It's a very good question. Of course, I'm just showing you a 2-dimensional table of things. Of course, it's much more complicated, and this is why I said so there are these different colors. So here, I'm just encoding who interacts with who, but I didn't tell you how strong they are interacting, how long they have to be exposed for the bonds to work. So there is lots of chemistry and physics going on there that I'm not telling you about. And this is, of course, the very important part of the design. But the blueprint is just a nice way to show who interacts with who. But there is a much more behind it that I'm not showing.
You said that if you control the blueprint, you will get much efficient self-replication there.
Mm-hmm.
So is the blueprint is fixed? I mean, by controlling means what? I mean.
So by controlling, I mean, designing the strengths of interactions between building blocks. The blueprint is just a scheme that we make on a computer, right? But what I really mean is, designing the DNA that goes onto the surface of the particle, like how long it is, how strong it is. So this is when I say design and control, I really mean the chemistry of the whole process. This is just giving us an idea of who should interact with who.
Thank you.
There's a question.
Hello. Thanks for the wonderful lecture and a lot of practical fun games too. One question is, right, is there any theoretical work on number of enzymes required for a particular structure to happen? Because it's like a combinatrix mathematical problem, right?
Okay, so that's a very good question. So what I can answer is that for the system that we know and control, right, where we have our beautiful spheres and we know the structures, we can really enumerate all the possibilities. Yeah, so the answer is yes. For a given system and given building blocks, I can enumerate the number of different enzymes under quotations, of course, because they're not real enzymes, but are artificial things that we would need to make something. So we can do that. Of course, we cannot do that for nature, but by doing it in these artificial systems, maybe we can learn how many there could be given some set of interactions. But yeah, in the artificial one, this is what we can do. This is the beauty of working with a system like this.
Okay, and the follow-up question is, right, which do you think came first, enzymes or the parents? I know it's a chicken egg problem, but I mean, what do you think happened in the primordial fourth or prehistoric fourth? I mean, the enzymes came first or?
Okay, so this is a very tough question, and actually in our Living Matter school, we recently had a discussion on this question. So the question is, how did the life started? So what are the origins of life? And there are two opposing hypotheses. One is that life started with DNA, somehow the spontaneous creation of a molecule like DNA or RNA. And the other hypothesis is that we actually had systems that were more enzymes catalyzing formation of other enzymes, so something more like a metabolism, and then the DNA came only later. Now, I am of the second opinion. I like more the metabolism first idea, but again, there are arguments for the first one as well. We don't have an answer to this question. There are good arguments for both sides. There are experiments that are trying to recreate some things, but we really don't know. And so I cannot answer honestly, but what I can say, I like more the metabolism first hypothesis.
In case of the blueprint for the octahedron
Yes.
replication, I guess we can collapse it to a network. I mean, the three-dimensional network. If you can collapse it to 2 dimensions, then you can get a better blueprint. That is I mean, you have 2 sets basically. I mean, once you have the network, then you can. That's what's happening, right? I mean, we didn't specify the shape. All we had is a network.
Okay.
So essentially, if we can collapse the 3-dimensional structure to a 2-dimensional network, then you have the thing basically. Then you can replicate the network. That's what we are doing, right? Because the geometric information is not there.
So, okay, so it is correct. If I just look at this 2-dimensional thing, the geometry information is missing.
Not there.
It's true. However,
so once basically you have the network, that's what we did. You are first growing the network. So once you squash down the octahedron to a network, then the geometry arises out of the energy and other constraints.
okay, but it's not as simple. If you just have a 2-dimensional, so if I have just a 2-dimensional blueprint, it doesn't guarantee me that it will work in 3 dimensions.
No, what I'm trying to say is that that's what we are doing in the sense that you have a network. Maybe you have one network on top. I mean, you have one triangle.
No, yeah. So this is true. This is a network in some sense. On mathematical sense, it's a network.
A tetrahedron on top and a tetrahedron on bottom, and both are linked to the. So if you can think of a graph.
It's a square by pyramid, actually, not a tetrahedron, but yeah, square by pyramid on top and the bottom. Exactly, they come together.
And they're connected by network. I mean, they're connected by the.
The base.
The plane,
yes.
The plane. So that can be represented as a 2-dimensional network.
Okay, so just to add, so the way that we try to find these solutions and enumerate things, we use graph theory all the time. So this is how we do it.
Yeah. Right.
Yeah.
It cannot be very stable, but the network is formed.
Yeah, sure, no problem.
Yeah. So no, this is absolutely true. So we start with using a graph theory a lot. So it's a 2-dimensional, and then later on we add the 3rd dimension, exactly 3rd-dimensional constraints, and prune the solutions that we found to the ones that we are interested in. Yes.
So, when you look at biological systems, structure formation is often associated with both consumption of energy and generation of entropy,
Yeah.
right? And listening to you now, I realize that I don't see those features explicitly present. So there are two possibilities. Either it's not necessary for structure formation spontaneously, which is self-replicating, etc., or it's hidden there somewhere.
Oh, it's hidden. Of course, it's hidden. You need, so biology, nature uses these energy storage molecules, the ATP molecules, all the time, everywhere. And I did not show you this. So there is definitely energy consumption that goes into this whole story that I'm not showing. But I couldn't, that would be very hard to demonstrate in the audience, so I skipped it. But yes, you're absolutely right. That's hidden in the whole thing.
Thank you, ma'am, for an enlightening talk. My question is environment playing a role in formations of these blocks and structures? Is it acting as a catalyst?
Okay, so that's a very important question. So we start from an environment we can control. And we first learn how to design these things so that we get what we want in an environment that we want control. And then the next step is, and this is what we dream about, that we're going to change the environment and see how things actually change and what happens and how do we make the system adapt to the changes or if it can adapt at all. But we start from a controlled environment.
Ma'am, what about the atmosphere environment?
So these are all either simulations or experiments, so it's all very, very fixed. So it's a jar of a solution at a certain temperature, or maybe there is some variation of temperature. So our atmosphere in that sense doesn't play any role. But this is how we design, because we are still at the very infancy of this whole thing. We're just trying to understand how to design these building blocks so they give us what they want. Changing the environment and actually doing something with the atmosphere is, I think, long in the future, but very important, of course.
Thank you. Thank you.
Yeah. Yeah, so I'm not showing you here, but you can imagine, just doing some things at the level of these DNA strands. And then that just changes everything, right?
Yeah, ma'am, it's an excellent lecture. I just want to ask whether you have not covered how the systems evolve, in the sense that how the blueprint is formed or how the catalysts are formed,
yeah.
Or what are the conditions or circumstances or compulsions for the thing to evolve? So how is it captured?
Yeah, so this is a tough one. I can say we are not there yet. So everything that we do is really just designing from the scratch. But this is really the dream, because we understand, and this is a very important point, and so thank you for asking this question, because we understand we have this. So we understand what are all the possibilities of these small structures that can happen, and now we are designing one, okay? And the dream is really that we are going to start evolving interactions and see how our structures go through this scheme. What do they become? What do they transform into something else? Do they become bigger? Do they become smaller? So we are not there yet, but we are not far from that. But this is really the dream of actually trying to evolve these blueprints and see what actually happens, how the system explores all the possibilities. I hope that answers your question.
Yeah, I just had a question on your comments on this early experiment, this Miller and Curie.
Yeah.
So where does that fit in here? Because that seemed more like a brute force type of situation which creates these early molecules, right? So
yeah. So the Miller experiment, right? So for those of us who don't know, so basically, many decades ago, scientists tried to recreate what they thought was the atmosphere of the prebiotic atmosphere of Earth. And so they had some conditions, and they put just building blocks in there. And they waited for some time in these conditions, and then they looked at what happens. And they found that some number of amino acids actually spontaneously formed. But they couldn't get anything more than that, exactly. Now, it doesn't come anywhere except that in the back of our minds, it's saying, hey, this can happen. Okay, so it is not easy, but it's not impossible. So if you just have different kinds of building blocks, we can imagine that these spontaneously happen. Now, I'm not working at this level of origin of life questions, so what conditions do we need? I assume I can just make anything, and I want to see how I can mimic properties of living things and how we can make them evolve. Yes?
So, if I understand correct, like this is what you were like the world is, I mean, the scientific community is able to actually experiment. Like you can see some motion when particles are put, this kind of motion. This is an actual photograph, I suppose, or simulation. This one?
Yes, this is an experiment.
This is, I mean, actually this happened. This is not a simulation, right?
No, not a simulation, not an experiment.
Okay,
they take these micron-sized particles, they put them in a jar, they put a solution in, and they look and see what happens.
And when it comes to replication, we have some results, something like this?
So in the replication, so this is, so I didn't have much time to go into those details. So what I can tell you is that, so they can efficiently make in experiments this, and this is what I showed you in this, hopefully in this movie. So they can make things self-assemble efficiently, okay? And there are some parts, sorry about going this very fast, but there are some parts of this blueprint that are realized in experiments, but the actual replication process required this thing that I sort of swept under the rug. I said, no, things separate at some point. So this is a hard thing to do. And even in the experiments that were tried in a different model system, they actually do the separation by hand. Okay. And so this is not really what it happens in nature. So it's hard to separate things. So in our system, we have a design in the simulation. We have a design of how this could happen. But this is still not something that's completely realized in experiments. There are some things that are missing. So they're all micron-sized particles, and we are all talking about micron-sized. And this is very important because this is also where biology is. These are nano and micron-sized objects because we really need temperature to help us things mix and move and do things. So we are at that scale.
We had several online viewers, so there's a question that should appear. We don't know where this person was in which part of the world, but there's a question there. So do you have a benchmark to compare against for the artificial building blocks we build? You mentioned there's a vast number of possibilities. Which one is a better function?
Ooh, this is a loaded question. Okay, again, so I cannot answer exactly because there are many designs, and there is also a question of what we are building. So do you have a benchmark to compare against? Okay, so we are definitely far away from what nature does. Okay, so we are at the level of just mimicking processes. So they're not as efficient, you know, the catalysis and replication, they're not as efficient as the biology does it, right? But it doesn't mean that it cannot be. So we are at for the moment, way, way below whatever exists in nature, but we are hope that we won't be for a long time. So that's, I think, the best I can answer for the things that we looked at so far, of course.
Thanks a lot for the wonderful talk. I had a question on, essentially, this kind of, again, a loaded question, two sides of the spectrum. We looked at some of the quorum sensing models, right? Essentially, enzymes, how each of the enzymes and the catalysts work. But if you look at virus, the way a virus works, it does not have, it's probably not even a cell. You know, if you consider that end of the spectrum, and also if you bring in a neocortex, a learning model into the system, how does that actually map to the evolution of artificial life, the way we are looking at it?
Okay. Okay, so I don't think I can say much. We are far from anything as complex as even as making a virus-like object. So yeah, we are just far from it. That's what I cannot tell more. I'm so sorry.
Hi, great talk. So I was wondering, in the self-replication, there are, this may be something someone alluded to earlier as well, but there could be two possible models. One is where you self-replicate the whole, when you have a large molecule of maybe, I don't know, 10 to the 10 number of atoms or building blocks, you could replicate the whole thing all at once, or you could replicate pieces of it and then those pieces assemble themselves, there could be several layers of that. Is that something that, is that probably how nature does it?
Well, nature has different things, but what is interesting from our perspective is to ask how things can happen, and then to see if there is just one solution, a few solutions, or restricted solutions. So to answer your question, so here we have a very simple object, right? It's basically all surface in some sense, and so it's easy just to glue things on. Once you have something in bulk, okay, you can still do the same thing. We've shown that you can do that. You have to do some tricks, but it cannot be a huge bulk. It really needs to, can go up to some depth, bulk, in the model system that we have. Which basically means that if you really want to have something much bigger, we have to have some hierarchy in how things are happening. So they are copied in some level, and then they come together at a different level in a different way. So I expect that at least in our system, this is going to be the best solution. Maybe there are other ones as well, but I suspect that this is the best one. And the nature has a lot of hierarchy, definitely, in itself. So it's not a surprise that this is one of the solutions.
And it would not necessarily be just one level of hierarchy, probably several levels, or?
Well, but I think in general they will have to. If we just imagine, mimicking some more complex living system, it has to be many, many, many levels, right? Because we start from DNA to the proteins that make this and that make tissues. I mean, it's just cells, tissues. It just has to be many levels of hierarchy.
Hi. I had a question about your experimental setups.
Yes.
Do you use, you said you do the experiment in the kind of a jar. That's just a simplified way of putting it, I know. Do you use a constant temperature, or do you have temperature gradients across the setup?
Very good question. So again, I'm a theorist, so the experiments are all done by my clever colleagues, so you should not let me in the lab. I think I demonstrated with our things that it's not clear I'm a good experimentalist. But there are some experiments that they do at fixed temperature, and some where they actually have a gradient to improve things, or they actually even oscillate between things. It really depends on what you do. And then thank you for this question, because there was another question of how things are done in experiments, and is this done? It's not done yet. However, a way to do it is to cycle temperature. So design these interactions in a special way that they have different strengths. And then the separation process can be done by increasing the temperature at some point and sort of things separate, but only these guys separate, so the original parents stay very strongly bound, so much, much stronger than the other ones. And then you reduce the temperature again, and then they make a copy.
Because each reaction would have a different optimum.
You can design these DNA strands so that they respond differently to temperature, exactly. So this would be a good solution. Thank you for the question. Okay.
Ma'am, I had a question. Like, we have computer viruses that self-replicate, right? Can we not study how these viruses really replicate, and can they ever evolve digitally to form something else?
Oh, so you're talking about computer viruses?
Yeah,
okay.
They self-replicate, right? They self-replicate and become bigger and bigger. Can we not study how they self-replicate, and can they ever evolve digitally to form something else?
There are people who are doing computer science that are very much interested in this and do this. I don't. I am more interested in material science point of view, so I don't replicate viruses, destroy things, I just replicate nice structures.
Okay, I had one more question. Does the molecules that we retain the properties of the particles? Does the molecules that we create from these particles,
I'm sorry, can you repeat from the beginning?
Does the molecules that we are forming, right?
Yes.
Do they retain the properties of the particles of which they are formed?
Oh, that's a good question. So here these particles that we imagine using and that are used in experiment are silica particles.
Yeah.
But you can imagine also making some magnetic particles of that size as well and using them, in which case your system can also have some magnetic properties. So yes, what the particles are can be very relevant for what you're building.
So they will retain the properties you are saying, right? But in real life, if you see, the actual form doesn't retain the properties, real properties.
Yes, but this is exactly what I was trying to say. We are looking at the artificial one. We want to explore different possibilities that we cannot see outside. And that's the exciting part. Yes, thank you.
Hi. So in the very beginning of your slides, you shown two lines. One was assembly line,
yes,
and the other one is the one we evolved more on.
Yeah.
So the question I was trying to put here is assembly line today, if something goes wrong somewhere, it's a top-down approach, and it breaks, the entire thing stops and doesn't go ahead. While at the bottom, what we have seen, things correct themselves and so on. So
yeah.
Can we build the engineering plants, the creation in this format?
Yeah, that would be the beautiful dream. I mean, this is why natural things are so fantastic, right? They can repair themselves. Like, imagine your computer breaks like mine does all the time, and it just self-repairs spontaneously. Wouldn't that be awesome? I would love that. I would pay for that. That would be the dream. Of course, correcting things, even to understand how nature does it, it's not a trivial process. So we are far from understanding how we can implement that in material science. But there are some systems already where chemists are cleverly designed things that can actually if they break apart, you just put them together and then they sort of heal. They don't correct themselves, they sort of heal. But yeah, that's the dream. That would be the hope, yes.
So, hi.
Oh, sorry.
Thank you for the wonderful talk. So I have no background in this particular area, but I was wondering, is there some kind of way to encode threshold or limits of self-replication in the blueprint or something like that? So we only need this much of amount of self-replication, but not beyond that.
This amount of self-replication, what do you mean
probably
exactly?
This many generations of self-replication, and is there some kind of decaying of some kind of parameter or something that can lead to not too much replication, but some substantial amount of that is
Okay, so at this stage, right, in the level of simulations and experiments, things stop when they use up all the material. So if there is no material anymore, it will just not happen. Now, if you can imagine that you use that to your advantage, if you can imagine that you want to build a very complex system where you just want to start from one thing, but you just want to have 20 copies, not more, and you just put that amount of material and you just have 20 copies. So you can imagine using that. I mean, what we do now, it just stops when we use up all the material that we've put in.
But what if you have substantial material, but you want to stop the... so I'm coming from some kind of... I work in artificial intelligence, so we have kind of theoretical scenarios where sometimes people fictionally think that our AIs can replicate themselves. So maybe there is some way of encoding. I was just curious. I have no background.
Yeah, okay, so there is, many decades ago, there was this concept of the gray goo. When people started thinking about these self-replicating things, there was this huge fear that things will start replicating uncontrollably and go out of the lab, and there will be this gray goo that covers the entire world. Yeah, so this is where we come back to our disclaimer. Whatever we do, we have to be very careful and keep in mind that we don't want even accidental things, bad things to happen. So what we do, we take care that this never happens. But yeah, I cannot guarantee that everyone does it.
Thank you.
Yeah.
So there are two questions here. So the last two probably. So.
Hey, nice talk, by the way.
during the bottom-up approach, as it happened in nature, it took like millions of years together. And now you talked about bringing simple particles whose properties we already know to a great extent, and make the bonds and form the octahedron and make them replicate. But as the evolution process takes place and the structures and the newly formed molecules, when you have to bring them together, don't you think a big amount of data is needed to, understand them and evolve? Because obviously we won't have so much time as nature had it. So is this data already available, or is this something you have to build in the process?
Okay, so it's a very good question. So if you want to imagine evolving these things actually and exploring the space, it's going to be hard, right? Because as systems become bigger, we need much more data. But this is where the beginning of my talk I said, so we are trying to understand some rules and regularities behind these processes. So what we hope is that by looking at these not so huge systems, so something that we can still handle with the modern computers, which really advanced in the last decade even, right? That we should be able to abstract some rules and regularities that will then help us be the explorer, with that vast map, explore things in a more controlled way. That at some point we won't have to look at all the possibilities, but we have rules in our heads, okay, which tell us, hey, you should go here to find something that's interesting. You don't have to go through all these things. So, I mean, of course, but this is the hope for the future. We are not there yet.
These rules, can they be deduced from the previous works as in material sciences, or do you think it's something that has to be newly found?
No, so I think that what we are doing in this approach is we are learning about rules. And although I didn't show you, I showed you basically none of them, there are rules that we are deducing even at in this level. And there will be more definitely, and there will be unexpected things definitely. But yeah, we are doing that slowly.
Thanks a lot.
Last question.
Thank you for the lovely talk. I just have two questions, part of which was asked by him already. First is, you described a lot about the experiments you conduct in the closed environment, and you talked about the effects of temperature that happens. My question is there also a factor of electric charge, and does that affect the binding of molecules in one way? The part two of the question is, as you mentioned, you are essentially trying to maybe build up a huge database of rules and maybe use machine learning later to do predictive analysis. Where do you see your whole thing in about 10 years from now? What is the picture? Thank you.
So the first question, it's a very important one as well. So what I didn't tell you basically anything about how these experiments can be done, but charge is of course very important, because if you can put in charged particles that then screen and prevent your DNA from binding. So for example, you can, if you want to prevent some things binding, and although they have complementary DNA, if you put some magnesium ions or something in, they can actually prevent things from binding, or other things can allow them bind much more strongly. So charge can help. So it's definitely, and this is where clever experimentalists and chemists actually rule, right? Without them, we would not be able to do this very nice. So this is very important. And your other question was about these database of rules, and yes, using machine learning to again find some rules. But can you repeat the question? Sorry. Oh, in 10 years from now. Ooh, that's a tough one. So the only thing is I can say I hope I will still be having fun. So I think this is the good way to stop.
Excellent.
Thank you very much.
Thank you, Zorana, for a wonderful talk. So a couple of announcements. First of all, there's Kaapi. So of course, after Kuriosity, go satiate coffee. The next Kaapi talk is on 27th May, and it will be delivered by Professor Joseph Samuel. And he's going to wonder something connections about steam engines and black holes and things like that. So come up. Thank you.